I would like to share with you our results on pure lab methodology for rapid point-of-care diagnosis of congenital Chagas disease using dried blood spots. Current diagnosis of congenital Chagas disease is based on direct parasite detection tests, such as the micromethod or microhematocrit, which is carried out in cord blood or peripheral or capillary blood of newborns or neonates during the first months of life, born to a seropositive woman. But these techniques have very low sensitivity and are dependent on the operator's skills. And then babies without a diagnosis must wait until they are 9th or 10th months of life for a serological confirmation, with a loss to follow up, mostly in those endemic regions due to economical and social constraints. So a high proportion of infants remain undiagnosed and thus untreated. Our aim is to optimize and validate molecular diagnostic tests appropriate for point-of-care detection of T.cruzi DNA, aiming to improve and expand the current diagnosis and treatment in newborns or neonates for seropositive women in line with the targets of the World Health Organization roadmap for primary health care. One of the potentially useful tools is the loop-mediated isothermal amplification technique, which is based on the isothermal amplification by a strand displacement polymerase of a highly repetitive satellite DNA sequence of the parasite. And the reagents are contained right inside the caps of the microtubes. This is a development of the Japanese company Eiken Chemical. After an incubation at 65 degrees during 40 minutes, and with the aid of calcein into the reaction, one can visualize the product of amplification by the naked eye or using blue LED lighting that is coupled to the heater bath provided by the manufacturer. The lamp has as advantage its high sensitivity, the use of very small volumes of blood. It's easy to perform. It allows even the analysis of dry blood spots. It has a fast processing and rapid reporting of results, and the kit does not need cold chain for transportation. Accordingly, we set up the conditions to be able to analyze artificial samples of blood spiked with different amounts of cultured parasites from different strains, using as solid support one-man paper or FTA cards, and our best results were obtained using FTA cards. When we prepare the DNA with the aid of an ultra-rapid purification system, also manufactured by Eiken Chemical, which allows to process FTA discs of around 6 millimeters into this series of tubes. And after 10 minutes of preparation, one can evaluate a single-stranded DNA directly into the strips containing the LAMP reagents. So after 1 hour of work, one can have the LAMP result. This methodology was transferred for prospective field validation to 9 sites in Bolivia, Paraguay, and Argentina, which are laboratories associated to maternities. We have trained the personnel at those maternities in the context of an international project composed by different institutions from Bolivia, Paraguay, Argentina, and Spain, and Japan and United States, with the funds are from Fundación Mundo Sano and from the Global Health Foundation. And we added to this work an external quality assurance program, sending proficiency testing panels to each laboratory. And we observed that working with the FDA cards, most of the operators from all the laboratories have very good coppa indexes in their work. So we expect that after finishing our field study, which is already undergone, we expect to provide evidences for a diagnosis policy change incorporating the LAMP for early point-of-care diagnosis of congenital Chagas disease.